Okay, so I think we can make a start. So, good afternoon, everyone, and welcome to the Palaeoverse Lecture Series. This lecture series is focused on software development and methodological approaches in paleontology, with the aim of promoting the use of new tools in the field and to give researchers a platform to showcase their work. With this series, we hope to shake up the usual type of lecture by having a mixed format with time dedicated to the presentation, demonstration, and discussion of the software or method presented. Now, typically these sessions will run for about 50 minutes in total. I will run this series throughout the year, generally on the last Thursday of each month, unless they overlap with the holidays. If you are interested in presenting a new method or software you've been developing or implementing, please feel free to reach out. We're always happy to hear from the community. So before moving on, just a quick few introductions. My name is Lewis Jones, and with me today I have Erin Dillon and Will Gertie. And the three of us are on the core team of Palaeoverse, an organization aiming to bring the paleobiological community together to share resources, reach agreed standards, and improve reproducibility in paleobiological research. If you want to know more about Palaeoverse, please check out our website and the GitHub organization, and the links are just being inserted into the chat below. So that's all from me for now. Erin will now give you some helpful tips and instructions for this webinar, and Will introduce this month's speaker. So thank you very much, and please enjoy the lecture. Over to you, Erin.
Awesome. Thank you so much, Lewis. So as Lewis mentioned, the format of the seminar will consist of a talk by our speaker, followed by about 10 to 15 minutes at the end that will be dedicated to Q&A and discussion. My fellow hosts and I will help field your questions and moderate this discussion. Please note that the seminar will be recorded and it will be made available on the Palaeoverse YouTube channel and on our website. However, we will stop recording before the Q&A period in case you would prefer not to be recorded. So before we get started, I'd like to quickly point out a couple of useful Zoom controls at the bottom of your screen. You'll see a Q&A button, and you can use this to submit questions to our speaker. And during the Q&A session, we'll either read out your question or invite you to turn on your microphone in case you'd like to ask it directly. There's a raise hand button. So alternatively, you can raise your hand and we'll call on you and invite you to turn on your microphone so you can ask your question. There's a chat function. So currently you can only chat with the hosts in case of technical difficulties. But during the Q&A period, we'll allow you to chat with other attendees. And then there is a closed captions button. So you can turn that on to view the transcript at any time. So lastly, I'd like to highlight a couple of ground rules. Our aim is to cultivate an open, respectful, and inclusive environment during this lecture series where we can all learn from each other. So with this in mind, we ask that you please be respectful and kind when engaging with our speaker and fellow attendees, particularly when asking questions and interacting with others in the chat. Any form of harassment or inappropriate behavior will be grounds for removal from the webinar platform. For more information, please refer to the Palaeoverse Code of Conduct, which can be found on our website. And Will is also putting a link in the chat now. So with that, I'll pass it over to Will to introduce today's speaker.
Thanks, Lewis and Erin, for getting us all up to speed on how the lecture series works. Today, I have the great honor of introducing our speaker, Dr. Allison Hsiang, an early career researcher in the Department of Geological Sciences at Stockholm University. Allison uses machine learning, high-throughput imaging, and statistical modeling to understand morphological evolution and macroevolutionary patterns and processes. She is particularly interested in bringing a next-generation approach to studying morphological evolution in deep time, for example, through automated big data collection. By bridging the fields of computer science and evolutionary biology, she aims to deliver a unique and creative perspective towards understanding evolution and the history of life on Earth. As part of this work, she has developed and worked on several software projects related to image segmentation and classification. So without further ado, we're really excited to hear from Allison today with a talk titled Automating Large-Scale Morphometric Data Generation Using AutoMorph and Deep Learning. Please join me in welcoming Dr. Hsiang to our virtual stage.
All right, thank you so much, Will, for the introduction. Let me just share my slides here. Okay, hopefully you can all see that. Yeah, so what I'm going to talk about today is mostly to do with this software package AutoMorph that I've worked on, and talking about how we can use automation and deep learning to automate, use automation and deep learning to build these large morphometric data sets of fossils. So as paleontologists, we're all interested in understanding biodiversity in deep time. And because of this deep time aspect, we're relatively limited in the types of data that we're able to collect. Specifically, we have to look at fossils. Now, in the last few years, there's been really leaps and bounds in the development of ancient DNA and how much ancient DNA we can sequence and how much we can analyze it. But the oldest ancient DNA that we have sequenced to date is about 2.4 million years old. So that's only about 0.06% of life history. So when we're talking about deep time, really for the vast majority of deep time, what we have to work with is rather morphology. Now, morphology is very complex and difficult to quantify. And so when we're working with fossils and we want to quantify morphology, it becomes a problem when we want to scale up from looking at just a few specimens to looking at hundreds to potentially thousands or hundreds of thousands of specimens. Because of the complexity of morphology, this scaling up process takes a lot of time, and then we have to deal with the complexity of actually measuring morphology. So my work is focused on, can we use automation and AI in order to sort of speed up this process and potentially also get at this complexity as well. So most of my work focuses on planktonic foraminifera. These are single-celled amoeboid protists. They construct an external test out of calcium carbonate, and they first appear in the fossil record about 200 million years ago. And the morphology and isotopic fractionation preserved in these calcium carbonate shells correlates with environmental parameters. So planktonic foraminifera are really important for doing things such as reconstructing climate patterns in the past. And they also have one of the most complete fossil records available because they are so cosmopolitan in their distribution and there's so many of them in the oceans. So when we're looking at these specimens, we want to be able to identify them to the species level. We want to measure them. If we're just, a single scientist looking at a few handful of specimens, it's easy to do this by hand. But as soon as we scale that up, again, this becomes something that nobody wants to do manually. So in order to deal with this problem, we developed this software package called AutoMorph. And AutoMorph, the basic idea is to be able to take an image that may have as many specimens on it as you want, to identify where in the image all your specimens are, extract out the individuals, and then extract out the outlines of these individuals. And from that, we can then get automatically a bunch of morphometric measurements, such as major and minor axis length. So this is just showing the types of measurements you can get. You can get aspect ratio, major, minor axis length, area, eccentricity, rugosity, perimeter. You can also extract out coordinates if you want to do outline analysis, for instance. And if you have Z-stacked images, you can also get an estimate of surface area and volume. And the advantage of this type of method is not only that it speeds things up a lot, but also it's much more reproducible than hand measurements. So what we have down here in the lower right-hand corner is an example of measuring the same specimens using AutoMorph 3 times versus measuring the same specimens by hand 3 times. And you can see that the spread for measuring things by hand is much wider. And so you can avoid that sort of issue by using an automated method like AutoMorph. So just some examples of things we've done with AutoMorph. So this is an example from a study that we did here at Stockholm University, led by Flor Formassen, who was a postdoc in the geology department. And we basically used AutoMorph to measure a bunch of planktonic foraminifera that were sampled from under summer sea ice in the Arctic Ocean. So the Stockholm University has a really big Arctic drilling program, and we went there and collected a bunch of forams, and then we were able to measure using AutoMorph about 15,000 specimens very quickly to look at what is actually going on in the distribution of these populations under the sea ice. Another study, this one led by Tirza Weitkamp, who is a graduate student in the geology department at Stockholm University, also on foraminifera. So she was focusing on an Arctic specialist species called Neogloboquadrina pachyderma. And she was interested in looking at what's going on in the water column in terms of what do these communities look like, and then what do these communities actually look like when they're preserved in the sediment after they die, and comparing those two. So using AutoMorph, we were able to measure 14,000 water column specimens and 11,000 sediment specimens to compare those. And she was also able to get some interesting insights into the reproductive cycle of these organisms. So this paper was just recently submitted, so hopefully we'll be able to get that published soon. So what we built AutoMorph for, so the examples that I just gave you are actually all with modern planktonic foraminifera. What we built AutoMorph for was to be able to not only automate the measurement of planktonic foraminifera, but also be able to identify them to the species level. And so for that, we had this project called Endless Forams, most beautiful. And what we did here is we combined two data sets. So one from the Yale Peabody Museum of Natural History, which contained about 60,000 individuals, and one from the Henry A. Buckley Collection of the Natural History Museum in London, which contained about 10,000 individuals. And so the Natural History Museum collection already had species labels associated with them, though we did double check these with two independent researchers. But the Yale Peabody Museum specimens, which came from Atlantic core top sites, from the about 35 of them, I believe. These we imaged, and so these didn't have associated species labels. So what we did is we gathered a group of about 20 taxonomic experts, and we had them basically look at all these images. We uploaded them to the citizen science platform called Zooniverse, and then we had them just identify as many individuals as they could. And an important thing to do when you're doing these sort of identification tasks is to make sure that the identifications you're getting are actually high quality. So what we did is we made sure that every single image was identified independently by 4 experts. And then in the end, we only kept the data where at least 3 out of 4 of the experts agreed on what the species in the image was. And so we ended up with a data set of about 20,000 individuals with these high quality species labels. So these are example plates of some of the species that are in this data set. You can go to endlessforams.org and download this data set and use it as you wish. But this shows you sort of the amount of intraspecific variation that exists within these species concepts. And so this is a really interesting data set also for sort of taxonomic training on these species concepts. But now that we had this data set with all these high quality species labels, what we then did was train a supervised machine learning model to be able to identify species of planktonic foraminifera using this data set. So this data set had 34 species of planktonic foraminifera in it, and we were able to train this model such that it could get accuracy of about 87.41%. And since then, since we published the Endless Forams paper, we have used a different model called a deep metric learning model, which has pushed this accuracy up to 92%. And we can compare this to the average human accuracies, which for this data set range from about 64 to 85% with an average of 71%. So you can see that by pooling together all this expertise of all of these researchers, we were able to create a model that's able to identify these species with higher accuracy than any individual expert could themselves. So that's another example of what you can do with AutoMorph. So the measurements, building these data sets that we can use to train supervised machine learning algorithms. But now let's move away from sort of these examples using modern foraminifera and talk instead about fossils, which is what we're all interested in. So this is a project that focused on the KPG mass extinction. This slide is here because when I give this talk to people who aren't paleontologists, I have to explain what the KPG mass extinction is, but I assume that this audience is pretty familiar, so I won't go into detail there. But what you may not know about the KPG mass extinction is that 90% of planktonic foraminifera went extinct at this boundary, in addition to all the other organisms that died out. And we really see an incredible amount of diversity that we've not matched since in planktonic foraminifera at the end of the Cretaceous, right before this extinction event. So I was interested in how do morphological patterns change in response to this mass extinction event within planktonic foraminifera in response to the collapse and recovery of the biological pump. And the biological pump is basically the system by which carbon is cycled between the atmosphere and ocean sediments via biologically mediated processes. And we know that when this extinction occurred, the biological pump collapsed, but there's also evidence to suggest that by 2 million years after the extinction event, the function of this pump has more or less recovered. But we don't see a concurrent recovery of the diversity in planktonic foraminifera. So I was interested in developing a high-resolution morphological record of planktonic foraminifera to look at what is actually going on in these communities morphologically in the wake of this extinction and in this recovery. So the data comes from this sort of famous site for planktonic foraminifera, the KPG boundary, called the Walrus Ridge, off of the west coast of the south of Africa. And this data set has, it's really great to focus on this data set for this morphological data because there's been a lot of work already on forams from this site reconstructing climate patterns. And so we can compare the climate patterns with the morphological patterns that we see. So the high resolution morphological record spans from about 500,000 years before the boundary to 2 million years after. So going from just before the extinction event to until we see this recovery of the biological pump. There's a total of 193 time slices with 772 slides, because for each time slice we have 4 different size fractions. And then we have an average of about 13,000 years between each time slice. And so this is the images of the automated microscope setup that I had that I used to image all of these slides. And then on the right side here, you can see an example of what one of these slides looked like. And what you can see is that I basically didn't want to do what is sort of the traditional thing to do with preparing these types of slides of forams, which is to hand pick the individuals and sort of arrange them on the slide to put them in a particular orientation and so on, because that takes a lot of time. And so I was really interested in, what can we actually do if we skip that, if we just basically pour the sample onto the slide, and then that allows you to prepare the slides much more quickly and also gives you a lot more individuals that you can sort of play with, around with statistically. So what that means, however, is that we have to be able to sort of pick out the individuals that we actually want to measure in a given circumstance. So for instance, you will have individuals that are broken or individuals that are fragmented, and we want to be able to pick those out, because if we're trying to get an idea of size distribution of the entire organism body size, we can't use fragments. So we want to have a way to sort of do this automatically. And we can do this using a type of model called instance segmentation model. These models that use deep learning to be able to simultaneously determine where something is in an image, so localization, while also being able to identify what this object is. So we'd had some previous studies where we had quite good performance of these models on planktonic foraminifera. So this on the left is a project by Nadia Arwanto, who was an undergraduate at Yale University. And she used one of these models to be able to pick out planktonic foraminifera on these micropaleontology slides, because when you have these slides, they have grids and numbers on them, and sort of traditional image processing algorithms aren't able to distinguish between an object that's lying directly on like a grid line or a number, because it can't deal with the lack of contrast there. But these instance segmentation models are able to do that. On the right here is another example by Marko Pavletić, who was a master's student at University of Gothenburg, who was able to train an instance segmentation model that was able to identify with very high accuracy different species of benthic foraminifera. So these types of models, so one type of instance segmentation model is a MASCAR CNN, which uses what's called a region proposal network. It's basically a combination of, so one part of the model deals with proposing parts of the images, region proposal, and the other part of the model deals with identifying what's in that region that's been proposed. And so the type of data that you need to give these types of instance segmentation models are basically pixel maps. So we have an image of forams here. This would be the type of training data that we would need to give to the model. So basically a map where we say, okay, identify all the pixels in this image that belong to the different classes that we're interested in discriminating between. So I was interested in discriminating between complete forams versus damaged, fragmented, and occluded forams. So unfortunately, for training these types of models, you do have to spend a lot of time building those types of training sets. So I labeled about 34,000 individual forams, those pixel maps that, like I showed on the previous slide, which sounds like a lot, but this data set, so if we're able to train a model that's able to identify these different objects by just labeling 34,000 individuals, that would be really great, because this data set, one single time slice has, for example, about 58,000 objects in it. So if we extrapolate across all 193 time slices, we're talking about a data set that has about 11 million objects in it. And so if we can get away with just labeling 34,000 individuals and get a model that's able to do the rest of these 11 million objects very quickly, then that's a good trade-off. So this is basically the current state of the model that I've trained using this training data. I'm still working on. So basically, the model is quite good already at picking out complete forams. It's having some trouble dealing with fragmented versus damaged forams. This is probably because the definition of these classes are a little bit variable. So I need to go in there and sort of try and clean that up. But as you can see, it's already doing quite well at being able to pick out all these different types of objects. And so what we're then able to do is to use this model to take one of these slides and automatically pick out which are the forams, which are damaged, fragmented, and occluded, separate them out. And once you've done that, then it's quite easy to take, just the forams that have been identified, the complete forams, and do measurements on them. So I took, for this particular slide, I took out all of the complete forams from the 150 micron size fraction, and I used AutoMorph to measure them. And to give you an idea of how much time that takes, it took about 6 minutes to measure these 672 objects, which obviously if you were doing it by hand, it would take a lot longer than 6 minutes. So that's really the advantage of these methods. So here you have in the bottom left-hand corner, island plots of the different size fractions and the different measurements, major, minor axis length, area, perimeter. And then you can also, as I said, AutoMorph can extract out these outlines, which you can then use to do outline analysis with. So that is it for the sort of research sciencey part of the talk. So now I guess I'll move on to the demonstration part. And for the demonstration part, I'm going to focus on AutoMorph. So this is a practical guide to AutoMorph, and I will talk about installation. I'll talk about the different modules that are available in AutoMorph, basic use case, basic usage in the command line, the control files that you use to run AutoMorph, the example data that will run, that I will run for the hands-on demonstration at the end. So definitely in terms of the feedback that I've gotten in terms of people who've tried to use AutoMorph, the most annoying part is trying to get it to install. So I've tried to make this as easy as possible, but you do need to have some experience with doing package management, Anaconda, and so on. And so the way you install AutoMorph is you first need to install Anaconda or Mamba. I would really recommend that you use Mamba instead of Anaconda. These are basically ways that you set up your Python environments so that you can get all of the packages that you need to run the code. Mamba is really a lot faster in my experience with solving environments. If you've ever had a situation where you've tried to set up a Conda environment and it takes potentially even hours, where the little thing is just spinning and it's saying solving environment, I'd really recommend you give Mamba a try. Usually that solves the problem. Once you set up once you've installed Anaconda or Mamba, you can install AutoMorph by going to GitHub and downloading AutoMorph. If you're on Mac or Linux, you can also run this optional install script. This basically just sort of creates symbolic links so that you can run AutoMorph from anywhere on your computer. And then once you've done that, you can set up your Conda environment. So when you download AutoMorph, there is a folder in there called env that has this file in it, this YAML file. This YAML file basically contains all the packages or the list of all the packages that you need to run AutoMorph. So when you create your conda environment, you can just give it this file and then it'll automatically download all the appropriate packages and set that all up for you. So you should theoretically be able to install everything you need with these 3 steps. But of course, package management with Python is always kind of a you can always run into surprises. If you do want to use AutoMorph and you ever run into any trouble, feel free to contact me and I'm happy to help out however I can. There is also a Docker version of AutoMorph. So Docker is basically a way to sort of package together these environments so that you don't have to deal with all this installation stuff. You can just run it directly using this Docker image. It's currently a private project, but if there's interest in this, let me know and I can give access to anyone who's interested. So AutoMorph has 5 modules. It has a segment module. This is a module that detects objects in the image and cuts out all the individual objects. There's a focus module that takes the output from segment. So segment can run on both Z-stacked images and sort of extended depth of focus images. So just a single image where that's already in focus. So focus was sort of built to, if you ran segment with Z-stacks, then you would run focus and then it would sort of create all these extended depth of field images of all the objects that segment had detected. So this is sort of something that we built because we needed it for our particular pipeline, but it's probably something that's not super relevant for the more modern pipelines. Run2D-morph is a module that when you have your images that have your individual objects in them, Run2D-morph will do the outline extraction and then also do all the automated morphometric measurements. Run3D-morph is a module that takes, if you have Z-stacked images, then this is the module that extracts the semi-3D data and gives you the estimates of surface area and volume. And then there's this predict module, which is basically only relevant if you're working with planktonic foraminifera. What predict does is it takes the model weights from our endless forams model. And if you give it new images of planktonic foraminifera, it can predict what species is in that image. So for today, I'll focus primarily on segment and Run2D-morph because I think this is these are the relevant ones for what I think is the basic use case for people who might be interested in using AutoMorph, which is basically that you have an image that has lots of specimens on it, and you want to run segment to be able to determine where all those individual objects are and get these individual specimen images, and then run Run2D-morph on these images so that you can automatically get these measurements and outlines. So the basic usage of AutoMorph is pretty simple. It's basically just in your command line, you give it the module name and then you give it the path to the control file for your current run. The relative paths are also okay. So if you're in the folder where your control file is, you don't have to give it the full path. You can just tell it point directly to that file. So it's important now that we talk about what these control files actually are. So these are basically just text files that give AutoMorph the parameters for the run that you're trying to do. So this is the control file for segment. And basically what we have here is, so these top 3, the directory, the output, or mode, as you can see, it says these are required. You have to tell AutoMorph these 3 things or it will not run. The directory is basically the directory that has the images that you want to process in them. So this can be a directory that has 1 image in it, multiple images, Z-stacked images. It can also be a directory that has directories that have images in them. You'll have to change some of the other parameters depending on which one it is, but we'll get to that. Output is the basically the directory that you want to output your results to. And then mode, the options are between sample and final. So sample mode is what you run when you're just starting out and you want to test out different threshold values to determine basically what is the best threshold value for determining where your objects are in the image. And I'll show an example of that soon. So then we move down to this block. So the single plane option is basically, I was saying that AutoMorph can deal with Z-stacked images and focused images. So if you have just focused images and not Z-stacks, then you would say single plane is true. Single dir, this is when this input directory that you've given AutoMorph, does it have multiple subdirectories in it or not? Include CSV, this is something that some microscopes, for instance, can, when they take Z-stacked images, they will also output a CSV file that contains the 3D information associated with that image. So if you have that, then AutoMorph can simultaneously, while it's determining where all the objects are, it can also crop out that CSV file at the same time. So then basically you would be isolating both the objects in terms of the image of the object and also the 3D data associated with that object. Transpose CSV is basically sometimes the output from these microscopes is the wrong way around. So you can switch that with this option. And then param file, this is relevant if you have multiple images or multiple directories. So basically, if you have multiple images or directories, you can tell AutoMorph that you want to use different parameter values for all of these files or directories if you want. So you can basically give it a CSV file that has all of the parameters specified. So then this is useful if you have multiple images that need unique parameters for processing. Pixel size X and Y, this is basically a conversion factor for converting between whatever your measurements are and pixels, like microns per pixel. This is generally something that's output by the microscope that you're using, or also if you have like scale bars, you can measure the length of the scale bar in image J or something to determine this. Then the threshold value, as I mentioned, so in sample mode, you can test different threshold values all at once, right? So here we've specified that we want to test threshold values between 0.2 and 0.5 by with a step between each threshold value of 0.01. And this can make obviously a very huge difference. So for example, here we have an image where on the left side, we've used a threshold value of 0.3 and on the right side, a threshold value of 0.5. And you can see that it really makes a huge difference in, how many objects AutoMorph is detecting. And then you can also control the minimum and maximum size of objects. So this can be helpful, for instance, if you have an image that has a lot of noise in it and you know that noise is quite small compared to the objects that you're actually looking for, then you can use this minimum and maximum size to sort of discard things that you are pretty sure are not actual objects. Yeah, the rest of these we can kind of skip. They're mostly metadata thing. This box thickness is, when you draw these red boxes on these preview images, how thick do you want them to be? So that kind of depends on the size of the images you're giving to AutoMorph. So then we'll see sort of this in action when I do the hands-on demonstration. Now for run 2D morph, control files looks like this. So it's quite similar. You have your input directory with your images. Input extension is what type, what extension the images you want to process are, a sample ID, just a name, what directory you want to output your results to. Pixel size X and Y is the same. So some of these are more interesting. So measure mode is basically when you don't need AutoMorph to sort of detect where the outlines are in your objects. And when this is relevant is, for instance, when I have these like binary images where you already know where the objects are. So it's very trivial to determine where the outlines of the objects are. You don't need AutoMorph to sort of run these filters to figure out where the outlines are. Then you can use measure mode equals true, and then AutoMorph will just measure the things you've got there. Get coordinates is if you want outline coordinates. Draw aspect ratio is if you say yes, then you'll get these images output of basically the box that AutoMorph uses to determine the aspect ratios of your objects. Adaptive thresholding is something that can potentially be useful if you're looking at an image that has very different brightness values across the entire image. So basically, if you use, if one side of the image is much darker than the other side of the image and use the same threshold value all throughout, then you're going to lose a lot of information. So what adaptive thresholding tries to do is it's an algorithm that sort of is able to adapt the threshold value based on sort of the surrounding environment So it can help when you have images that have a lot of variation in brightness and contrast. And then save intermediates. If you turn this on, what AutoMorph will do is it'll basically save all of these individual outputs. So basically run 2D morph, it runs all these filters to determine the outline of the object, and it will save all of these intermediate images if you turn save intermediates on. And this is really helpful for if you want to troubleshoot. For instance, if you're not getting very good outlines and you're not really sure how you should change the different threshold values and parameter values, then you can save the intermediates and then look at these and then try to figure out where the problem is. So adjusting these parameters are what the rest of these things are, which I won't go into detail about, but if you have any questions, let me know. So the example data that I'll now use for the demonstration is from this Yale Peabody Museum dataset that we imaged for endless forams and which we describe in this paper, the scientific data. So basically, we're going to look at this image right here and run a segment and run 2D morph on this image. So we'll do that now. So I'm going to stop the slides and share. Okay. Okay. So hopefully you can see my terminal window. So I already have AutoMorph installed and the Conda environment installed and everything, but I can just show you slow. Okay, so the environment that we want to use is this AutoMorph environment here. So we can activate that. Okay, so the data that I'm looking at is in this folder. So basically this plane 31 resize, that's the image that I showed you on the previous slide. So what we're going to do is we're going to run segment on this image to get out all our individual objects. And then once we've done that, we can run 2D morph on it to get all the measurements. So I have here, what we're first going to do is to try a sample run. So this is where we're testing out the different threshold values. So let's just look at this control file. So here we have our directory with the images where we want to save it. We're in sample mode. So this image is just a single focused image, not a Z-stack. So we're just going to say single deer is true. Single plane is true. It's a single deer. We don't have subdirectories to go into. The rest of these we can leave. If you leave anything blank, except for these top three, which are required, if you leave anything else blank, then AutoMorph will sort of use the default values. These you can look up in the code. So for every module, there's a file called settings.py. If you go in there, you can see what all the default values are for these parameters. So we're going to test a threshold of 0.10 to 0.15 with a step interval of 0.01. So that's going to be 5 threshold values. So we can do that by just doing segment control Palaeoverse example.txt. Okay, so the first time you run this in a session, it takes a little bit of time to spin up. So I'm just going to go ahead and show you the results because I've already run this. So basically here, once this completes running, what it will give you is one image for every threshold value with the objects that it has identified using that threshold value. So here we have the threshold value of 0.1, 0.11, 0.12, 0.13, 0.14, 0.15. And we can see from this that the best threshold value for this particular image is going to be 0.12, because here, if we go to 0.11, we've got some really big boxes here, some noise that we don't want. If we go to 0.13, we lose this object here. So we want to use 0.12 in this case. Okay, so now you see that it's run here. When you run it, it will show you, what threshold value it's using and how many objects it's found and so on. Okay, So now that we've determined what the best threshold value is, we can run our final run using that threshold value of 0.12. So if we look at our control file, this is the final run, so we're in final mode. Everything else is the same, except that for the threshold value, we have this 0.12 threshold value. So now we run this. Okay, it's done. So we can go look at that. And so when you run in final mode, what will be outputted is there will be a text file with all your settings so that you know exactly what you did. There will be a preview file that has the full image with all the boxes drawn around the objects that AutoMorph has detected. And then there will be a folder called labeled. So these are all the objects AutoMorph has detected with this sort of metadata label attached to it. And then there will be a folder called unlabeled, which has all your individual images that Segment has found. Right. So now that we have this folder of individual images, we can now run 2D-morph on this folder. So if we look at the run 2D-morph control file, I've pointed it to this folder that you see on the left. These are TIFF images. We're going to output it to the same directory. Everything else we're going to leave default. I'll just point out here that let's draw the aspect ratio images so that you can see those, and also save intermediates so that we can see those as well. So then to run this, we just do run 2D-morph and then the path to the control file. And there it goes. Okay, so once it's done, we can look at the output. And the output will give you, again, the settings file that has all the settings that you've used for this run. It will give you then also a CSV file, and this is what contains all the measurements that you have for all these objects. So AutoMorph, for now, these are the measurements that it's able to give you: area, eccentricity, parameter, major, minor axis length, and so on. You can always either ask me or fork the code yourself to try and add more parameters, depending on what you need. But yeah, that's sort of the default. It will also give you a text file that has the list of all the objects where it failed to extract measurements. And then we have, so we asked it to draw aspect ratio, so that's the folder with all of these. We also have CSV files that have all the coordinates for outline analysis. We have, the outlines folder, which just has all the images. You can see that some of these, most of them are doing quite well, but some of them, like this one, for instance, it hasn't managed to get the entire object. So this is usually an issue with when you have an object where one part of it is darker or lighter than the other part of it. This means that you have to sort of play around with the threshold values, or maybe use adaptive thresholding to try and fix these problems. But when you have these problems, what you can do is then go to intermediates and then look at, so here's every step that AutoMorph is running to get this final outline. So for this one, for instance, you can see that there's this part on top that sort of disappears when we do thresholding between grayscale to black and white, which means that in our final outline, we're missing that part of the object. But because you can see these intermediates, that means you know where the problem is. So that means you have an idea of which parameters to adjust in your control file to try and fix this problem. So yeah, that's it for the demonstration and the talk. So I'm happy now to answer any questions.